live. Okay. One sec, we are going live. Okay, good. Perfect. Okay. So we are now live. So Thierry, I think I leave you the show. We are going live.
Yeah. So thank you, Caterina, for giving me the opportunity to invite some people from the neuroscience community. And today I have the honor to host in our CCB meeting Valeria D'Andrea. So she got a PhD in Rome from Sapienza, and during a period of her PhD, she was a visiting student also in Germany at the Max Planck Institute of Tübingen. And after that, she joined the Italian Institute of Technology, the neurocomputational group in what's the city?
Rovereto.
Rovereto. Okay, I was thinking about Trento. Yes, and actually, during that period, so it's around 2016, I guess, or just before, we met and we started collaborating on a project that we published together. And after that, we left IIT at some point, and she joined then the Fondazione Bruno Kessler in Trento. She joined the group of Complex Multilayer Network Lab. After that period, so after 2021, she joined the Health Emergency Center, which, as we can well remember, was quite important at that time. And during that period, she started to study also to work on a statistical and mathematical model of infections, disease transmission. And well, today actually she will tell us much more about network dynamics. So she will tell us about probably the activity she did before in the group of Complex Multilayer Network Lab. And I'm particularly happy as she is giving us a talk today, which is really moving across different sectors. In particular, she will tell us about reservoir computing, which is a paradigm of learning that is gaining quite a lot of interest in different communities, like also the neuromorphic community. And I think from the abstract that I read, actually, I guess it's a nice approach toward the study and a new way to deal with this type paradigm of computation, as she is actually putting much more biological constraints into these paradigms. And so I'm really happy to hear about this talk. And so I leave the floor to Valeria for the talk. As far as I remember, we have around 45 minutes, then we have time for questions.
Okay, thank you, Thierry. Thank you very much of all of you for inviting me at this area of the webinar. It's an honor for me to be here. And let me share the screen. So maybe I don't have the permission.
Yeah, Thierry, you are the host, so you can authorize Valeria.
I'll try again. Okay, here we are. So let me find, I think that. Okay, I hope that now you can see my
Yes,
screen.
now it works. Now it works.
Okay, okay, but maybe it's not the right slide. Okay. So again, thank you again for inviting me. It's a pleasure to be here and to present to this audience our one of our last works about the reservoir computing method and the interplay between the structure, the network structure in the reservoir and the function in connectome-inspired reservoir computing. The reservoir computing is a state-of-the-art algorithm of machine learning methods, and it is inserted in a broader background. And I would like here to just sketch all the background in machine learning for the ones in the audience that maybe don't know this kind of framework very well. And but first of all, let me say that this kind of historical context that I'm going to present is not exhaustive, and above all, I choose to describe only the algorithm in the machine learning that are in some sense are related to the biology, that are inspired by the biology and by the structure of the brain. Let me start with the first example of machine learning, that is the perceptron that was invented in 1943 by McCulloch and Pitt, and it combined in a linear way inputs in a neuron, and it produces an output that, when traded, when the perceptron is trained, can learn the nature of the different input. This is a very preliminary method because it can only learn some kind of input, some data set that can be linear. Divided. It cannot manage non-linearity in the input. In the late 40s, so about 1948, a psychologist that is called Donald Hebb invented the so-called Hebbian learning rules that was inspired by brain plasticity, and attest that the connection between two neurons, so the learning is in some sense derived by the activity of the two different units and are encoded in the weight between these units. And inspired by this kind of learning, in 1948, Turing built this is a machine that is called B-type machine, inspired to this kind of learning. The fourth architecture that we find in machine learning, a little more sophisticated, is called the multi-layer perceptron, or a kind of feed-forward network. In this kind of network, we have different layers. And usually the kind of input that is learned are only spatial features, so usually are images. So it doesn't manage any dynamical system, any temporal features. In this case, the input is some features of the input are passed to the first layer in the architecture, in the network, and the activation function of each neuron, it depends on the weights that must be trained from that unit, and then the activation function is conveyed to the other neuron in the architecture in order to learn the different features of the input. With this kind of architecture, we can manage non-linearity of the input, but still we cannot yet manage the temporal future of the input. This kind of temporal meaning, this kind of temporal learning was achieved some years later with a different kind of architecture in machine learning that are called recurrent neural network, in which basically there is not only a forward architecture, so the input is not only conveyed in a forward way to the other layers, but the neurons, the unit, they are more forward in the architecture, can send again the input to the neurons that are earlier in the structure. In this way, this kind of feedback mechanism is very important, because in this way you can encode the temporal properties of the input in this kind of structure. And so you can also have a sort of memory of the input. You can store you can in an efficient way learn dynamical system with this kind of architecture. I also put here another kind of recurrent neural network that are a little bit different, that are called energy-based recurrent neural network. In this kind of architecture, you have the connection between the nodes. The units are symmetrical, and the learning is achieved by a sort of minimization of global energy of the network. An example that I put here is the famous Hopfield network. And in this case, the memory is stored in the connection between the different nodes using an Hebbian rule that I introduced before. So all these architectures are very, are very efficient in for describing also dynamical systems. So also the temporal future of the input, but still the training of the network that is finding the right weights of the nodes that are optimal to learn that particular task are still a problem. Is a very slow process, and not always you can reach the optimal learning of the network. And so we arrived in like 20 years ago to the reservoir computing kind of architecture. We have in 2002 both Jaeger and Maas are two different scientists that built this kind of architecture. Jaeger invented the so-called echo-state network that are more based on machine learning method. Instead, Wolfgang Maas write about the liquid state machine that instead are more inspired to biological network. But the principle that are behind this kind of architecture are the same. So in fact, later they were called under the same name of reservoir computing, which is the novelty of this approach. This approach use recurrent neural network as the reservoir. They are sparse and nonlinear neurons, but the novelty is that only the output layer are trained to learn the input. Instead, the weights of the neuron inside the reservoir are fixed and usually are assigned in a random way. But just by training only the output layer, you can train the network in a very fast way and in a very efficient way. So yeah, just let me say that one of the goal of the project of our project is to understand which is the role of the structure of the network inside the reservoir, because as I said, usually the weights and the connection are assigned in a random way. Instead, we asked, but is not a new concept. Jaeger itself wondered that maybe the structure internal to the reservoir can have an important effect on the learning of the task that you are performing. So from this kind, from the machine learning perspective, we are asking which is the role of the structure. But the kind of question we want to answer has also an important meaning from a neuroscience perspective. Instead, let me skip. Let me just introduce some concepts that are very general in neuroscience, but I will go very briefly. We know from some years that cognition, I wrote here, cognition is embedded in a multiscale structure of the brain. The connectomes, they are the map of the connection in the brain, were analyzed with the tools from the network theory. And we saw that those kind of networks, brain networks, have some features that are typical of complex structures. For example, they present, they have this small word architecture, or we can see in the real brain connectome, we can see the presence of hubs that are nodes that have a number of links that are much greatest from the average of the links, of the number of links. And also we can see we can say that brain network as a modular structure. Those are all features that belongs to a complex network and that were studied in real brain connectomes. But also we see that we can say that the structural properties of the connectomes that can be both structural and functional have an important role in the function of the brain. For example, we saw that in healthy subject, we saw that the connectome, the brain connection, the connection in the brain depends on, for example, the fact that you have performed a task before or not. Or also in healthy subject, we saw that the structure percent. So yes, there is depends, for example, on age and on the type of the task that are required. But also we see that exists some difference in the brain structure between healthy subject and subject with a series of disease, for example, Alzheimer's disease or epilepsy and many other diseases. So the structure of the connectome, and this is a real, not only at the whole brain level, but also at the small scale. The structure has an important role in the functioning of the brain, and there is a neural plasticity that affects the structure. And yes, now we arrive at the piece of work that we addressed with this paper, but is a larger project. We want to study which are the relationship between the structure and the function in a real brain connectomes. And as I said before, this study has a double purpose. From a machine learning perspective, it is useful to see if we can improve the performance of the machine learning. Instead, from a neuroscience perspective, it can be used to better understand how the cognition is structured in the brain. So we use a lot of structure. I will introduce it. We used a series of real brain connectomes, but also we tested our result on a series of synthetic connectomes from some network model. We also checked if the nature of the signal that we want to predict has an effect on the prediction. We choose two kinds of signal, but stochastic and deterministic. And we also test how the dynamical representation of the input is affected by the structure in the connectome. So, as I said before, we selected as a reservoir a set of real brain connectomes, and we try to predict two kind of noise, two kind of signal, two stochastic signal, a white noise and a fractional Brownian noise, and also two stochastic but deterministic signals, so a Mackey glass and a Kedemap. And we checked the performance of the reservoir in a task which is very simple, is a one-head prediction task. And then we measure the performance of the reservoir, in this case with the mean square error between the input signal and the output. Here I put the activation state matrix, which is a very important matrix, which contains all the dynamics of all the neurons in the reservoir, and will be very useful in a while to describe how the input is encoded in the structure of the reservoir. About the real connectomes, here we use 45 real connectomes that belongs to 8 different species. We have connectomes from the Chonenchesinalis to the human, and they are very different. For example, they span from this graph, you can see they span more than 2 order of magnitude in the number of nodes, and more than 3 order of magnitude in the numbers of the links of the connection of the links of the reservoir. And also they are very different according to the network modularity. So the presence of different, but we will see later, the presence of different communities in the network. We also checked to check if the results that we obtain with the biological, the real connectomes are robust. We checked our results also on 3 different trinomial models from the network theories. The first that preserve some of the features of the real brain ones. In this case, we choose an Erdős model that in this case, it preserves just the average degree, so the average numbers of links for each nodes of the real connectons. We also choose a configuration model, and that in this case, it preserves the degree distribution of the real connectons. And also a stochastic block model that in this case, that is more complex. We choose three different null model with increasing complexity. The most complex is the stochastic block model that preserves both the degree distribution of the real connectons and the block structures. So let me skip to some results of this study. So let me start from the real connectons. We took the real connectons and put it in the reservoir and check the performance of the reservoir. What we found is that the prediction error depends on, for example, increase in a stochastic signal, the error increase as the number of the nodes of the connectome increase. Instead, we see the opposite effect, so the performance is greater for more number of neurons for the stochastic deterministic signal. In this plot, the color of each dot label the number, not the number of the nodes in the connectome, but the numbers of neurons in the brain's organism from which the connectome is taken. So this kind of trend was found not only for the number of nodes of the connectome, but also for a set of different features. For example, the average degree and the modularity of the connectome, so the presence of the presence of different modules in the real brain connectomes, and also an important measure that is the number of independent vector state. This measure is basically the rank of the covariance matrix of the matrix of activation state that I presented before. And then that is an important measure because it encodes the number of nodes in the reservoir that has independent signals. So in some sense, it encodes the number of the dimensionality of the reservoir that is effectively used to learn the input. But the trend is the same. In a significant way, you see that the performance increase. In this case, when the error increase with the number of independent vector states for stochastic signal, and the trend is the opposite for deterministic signal. So we also checked our result, as I said before, against the three different models. In this case, so we checked the performance of the reservoir against the Erdo-Chouini, the configuration model, and the stochastic block model. In this case, to perform, to encode this comparison, we use the zeta score. And in this case, positive zeta scores means that the quantity in the synthetic network are greater than in the real ones. In this case, we see that for all the kind of signal, and we see that for the synthetic network, the number of independent vector state is greater in the synthetic network with respect to the real one. We also see another result that Yes, so the number independent vector state is greatest. That if we fix the organism, the performance, the difference between the real and the synthetic network increase when we increase the complexity of the synthetic model that we are testing. And we also see another kind of result that if we fixed the synthetic network, we see that the difference between the synthetic and the real network increase has the number of neuron of the brain increase. So in this case, for example, we obtained the greatest difference between synthetic and real connectomes for human subject. So because we find that the number of dependent vector state is the greatest in synthetic network, we would expect the same kind of trend also for the performance of the reservoir. But that is not true, and here we found a sort of counterintuitive result, because if we see that the performance so if the arrows in this case in the deterministic signal are lower in the synthetic structure than the real one, so this is expected in some sense, we see that for stochastic signal we have a opposite trend. Instead, the real connectomes perform better in that case. Anyway, the finding that we have about the structure, so that the complex structures are the ones that best predicted the signal are still found, and we see, for example, in this case that also in this case the stochastic block model is the model that have the best performance. So maybe I was too fast, but I would like just here to say something about the ongoing research, because as I said before, this is a wider project that was. I mean, this in turn of the Comune lab, the lab that now is in Padua, that is led by Manuel Domenico. And of course, other people. At this step, I mean, we are trying to better understand our results on the real and the synthetic connectomes. And in particular, we are investigating the relationship between the performance in the reservoir and some network information theory measures, for example, the spectral entropy, but also the description length or the free energy of the network, because we can leverage on a series of studies that were performed in our lab previously, that are grounded on statistical field theory of information dynamics. And we also can exploit. We also have a huge data set that in this case is provided by the lab of Maurizio Cordetta in Padua. And so we want to apply our analysis not just to a series of connectome from different spaces, but to a series of connectome of both healthy subject and subject that with the strokes. they performed a sort of a longitudinal study at 3 weeks, 3 months, and 1 year. And so this is a huge data set at a different resolution, so a different parcellation that we can use for our analysis. And that's all. I don't know if I was too fast, but yes, I hope that you have questions, and yes, I'm available to answer you. Thank you very much.
So, thank you, Valeria. I think there are certainly some questions. At least I have some, but let's see if there is someone who has a question in the audience. Otherwise, I'm going to start. So I missed one point. Actually, I could interrupt you at the beginning, but I missed how you train and how you test the reservoir. So what is kind of signal, because you're trying to predict a temporal series.
Yeah.
And so what's the input? Sorry, the training, the test, the training. Yes, the training.
So I mean, we use a part of the signal,
okay.
dynamical signal, as a training. Of course, as usual, a part of the signal as a test. Okay. But the training, the important things is that the training in the reservoir computing is done only on the output weights. So we just trained the output weights.
Okay. And just as a curiosity, it's more technical stuff. Do you also perform some kind of cross validation? I mean, I mean, this type of training is performed like the standard way
yeah, we just I don't remember exactly how much signal we took for test and training, but we repeat our analysis, I think 100 times, but several times. So that is not a cross validation in the usual sense, but yes, we repeated this kind of training a lot of times. So.
And again, it's more maybe technical. You mentioned the Hagman connectome, the human connectome. Is that the one? I guess it is the one published years ago about stroke modeling. I think it's among the connectome you were showing us.
At the end of the presentation, you mean?
Not at the end, actually. It was in the middle, I would say. Maybe here, and just some slides after this.
Here?
Among the different human connectome you tested, actually.
Okay. Yeah, maybe there is something here in. Yeah,
here. Yeah.
Okay. There are connectomes that you can find in literature. There were from previous paper. I don't know if yesterday they published something, but yeah.
I guess it's around 9,000 different areas, right?
Okay. Yeah, there are, but we choose to select a wide set of structure in order to investigate which are the role of the different number of units in the connectome, but also we wanted to test if different species perform the task. It's a very simple task, of course. A lot of things can be done and will be done with different type of tasks that, for example, involve memory or some complex task that the human can perform. But in this case, we focused only on a simple task, a prediction task that can be probably be done also by simple kind of organism like worms or similar.
And around this slide, or maybe the next one, you you briefly mentioned the fact that the human connectome was superior to other connectomes. Not sure. I probably.
Yeah, the result is that not only the number, because of course one can imagine that then if you put a greater number of neurons in the reservoir, the performance will be better. But the some of interesting results that we found here is that the human, you can find the best prediction in humans, even if for the human connectomes we don't have the maximum number of nodes of the connectomes. We know that they have the greatest number of neurons in the brain, but the connectomes that we check doesn't have the greatest number of nodes. So this is very interesting because we cannot explain it. That's the point. In this kind of study, we found a lot of results. But the point is to explain, to ground them well with other kind of measure that I said in the end of the talk. Mm-hmm. So yeah.
Okay. So are there any specific features that are retained by biological networks that are more relevant than others?
Yeah, the block structure. We know that brain has the so-called hierarchical block structure. So it is hierarchical and modular in the same time. So that kind of architecture, that are complex architectures, are very important for the prediction in this case in the reservoir, but yes, this kind of structure are present in the brain and of course have a lot of importance.
Okay,
so hierarchical block structure.
Okay, perfect. So we have other questions from Bastian.
Yeah, thanks for that great and interesting talk. I was a bit confused or wondered if you have some intuition why some models perform better or worse than others on white noise, because you would expect that there's like no ability to predict anything anyways, and so that there shouldn't be a difference. At least I would have assumed that.
You mean about the white noise?
Yes.
Yeah, it is just to know, for example, how, if of course, if you the fractional Brownian is a sort of is different because you have a sort of correlation between the time point. But we also checked the white noise to see if there is some sort of how much the input and the output just diverge. So another things that we can do maybe in the future is just to normalize all the measure to the white noise one, just to see how it can be related to the white noise. Yeah, I imagine that try to predict the white noise is a sort of confusing, but we select this kind of signal just to know how the reservoir is performing.
Mm-hmm. Do you know why some network structures perform better or worse on white noise?
No, for the white noise, not yet. That's the point.
Okay, thanks.
Okay.
Any other question? Curiosity, small things.
Hello, am I audible?
Yeah, please.
Yeah, so actually, thank you very much for this very insightful talk. I just wanted to ask one thing. Are there any algorithmic biases in terms of finding modularities or looking from the algorithmic perspective?
Mm-hmm. Sorry, about the modularity?
So I was just asking about the algorithmic biases in terms of identifying modularity or the related network stuff.
So you are asking, sorry, because I don't know if I get it well. You are asking me if the modularity has an effect on.
So I was just asking, so most of the time networks, from network structure and topological features, we are somewhat very sure of its nature, and we can define its dynamics. So I was just asking, is it purely algorithmic driven, or we have some biases in terms of algorithm? Regarding computation, I mean, if we want to predict some structure from the input thing, right? So do we have a sole dependency on algorithm that is like
so
bias?
You wanted to predict the structure from the. You want to predict the structure from.
So I want to map the dynamics from the structure, right? So sometimes we have different algorithm, right? So there may be some biases with respect to those algorithms, right? So I just wanted to ask if there those things are covered in some recent work or not. Okay. I worked in the domain of complex system, but I'm new to neurological application.
Yeah,
so that is what I was asking.
I think that if I got it well, the questions, it's possible to understand something on the structure of the network from which you are sampling the signal. That is possible, and it are actually we are actually doing it with a sort of a Kuyamoto model. So we are simulating a Kuyamoto model on network, and we are able to understand something on the connection between the nodes. So we are able to say something on the structure of the network that we are analyzing just by watching at the signal in the node of the Kuyamoto of the network. So yes, it could be done. We are actually doing it in our lab, and yeah, I hope to have soon some results to show.
Yeah, so I just meant that we have different types of community detection algorithms. So in that sense, it depends upon the requirement. So we can have different algorithm for finding communities. So I was just asking, how about those biases if you use neurological data? So we may have one type of result by using one algorithm and another type of result by using another algorithm. So how will you rely on this outcome?
When you say algorithm, you mean different machine learning algorithm or?
Yeah. Different application. I mean, we have different algorithm from machine learning in order to identify communities, right?
Yeah.
So if we fed neural data into that, so we may have two different types of result, maybe very close to each other. So how to deal with those biases? I mean, in terms of interpretation.
I don't know. Actually, we don't know yet. I mean, we are just studying which is the interpretation. We just focus on a simple algorithm because the reservoir is the simple one and can be easily trained. It is the more biological one. So it was natural to use this algorithm to put the connectome inside and watch for the. Of course, you can do this with the other kind of algorithm, but I don't know how could be done in this sense with a biological meaning. Of course, you can of course done this with a different kind of network. But if you want to know something about the biological meaning, I think that the reservoir is the most useful.
Yeah, so this is what which I was mentioning. We have biological meaning, right? So with one algorithm, we have one set of biological interpretation, and with another algorithm, we may have slightly different.
I don't know if the biological interpretation, I see at the different algorithm, if we are talking about machine learning algorithm, I see at them as an evolution. I mean, of course, you can do this with a feed-forward network, for example, but you lose a lot of the nature of the thing that you want to predict. Actually, with a feed-forward, as I said before, you cannot predict anything that has a temporal structure. So since we are interested in something in the brain that must to perform some temporal task, we are interested in some algorithm that can do this.
All right, thank you very much.
Do we have other questions from the audience? Otherwise, I think we can close it today, even with 10 minutes in advance. And so, yes, it was quick, but I think it was quite effective. And so we thank everybody for participating to this meeting, and especially Valeria for giving this talk.
Thank you.
And okay, so see you next time in April for the next CC&B talk.
Bye.
And goodbye to everybody.
Bye. Thank you.
Goodbye. Thank you. Goodbye.